So I'm going to introduce the speakers and then take questions or have some question myself. So the first person I'm going to introduce is Dr. Rachael Tarlinton. She's is a professor, associate professor at the University of Nottingham. So she uses technologies such as whole genome sequencing on host and pathogen to study epidemiology, to study how viruses associate with host. She's been interested in the host range of coronaviruses in European wildlife, host genome integration of retroviruses in like some more ordinary animals like cats and rodents, but also some more exotic like koalas because she's Australian, and she's also interested in emerging vector-borne diseases in domesticated animals such as ruminants. And so she studies genome epidemiology and also social sciences approaches that all come together to provide option for control of viral disease in animals and by domesticated animals and their owners. So I hope this gives you justice, and the floor is yours.
There we go. So that should be it. So
Brilliant.
after that lovely introduction from Julia, here is what I thought we'd do today is a bit of a whistle-stop tour through where we're up to with European wildlife and coronaviruses, because a lot has happened in the last 5 years. So I skipped most of the intro coronavirus bit for this audience, but just a reminder, because this is important when talking about cross-species transmission, is the main ones we are talking about as risk to mammals are generally ones from the alpha and beta coronavirus group. So SARS, MERS, etc. The recent human cross transmissions are mostly in the beta coronavirus group, and the gamma and the deltas are primarily associated with birds, and spillovers into mammals are quite rare as far as we know at the moment. So, to all intents and purposes, we're talking about alpha and beta coronaviruses today. So that's this in a different form. And the other thing to remember here is that uh, what we know about this is very much limited by the species we have studied. In species where there has actually been an intensive amount of work, which is really just humans and pigs, we know they carry multiple coronaviruses from multiple genera. So we've got at least 6, but they're different ones in humans and similarly at least 6 in pigs. And probably this goes on a lot more. We know we have at least 3 different variants of alpha and beta coronaviruses in dogs, but for most species, we just don't look very much, so we just don't know. There are most of the work that's been done has focused on a limited range of economically important species, and most of the wildlife work has looked at either mostly at bats, occasionally at rodents. But the little bit we do know demonstrates that these things cross the species barrier an awful lot. They do move around a lot, and they're not often not terribly host specific. So it's a little diagram that's not from my paper, but it's a nice diagram of the variety of ways in which the various human coronaviruses are thought to have ended up in us. So I'm going to just cover kind of the major mammal groups we have in Europe, and in the UK, of course, because we're based in the UK. So bats. So this little map here is from some collaborators in Portugal, and it's just a summary paper of all the different types of coronaviruses and all the different bat species that have been published in Europe. And basically, it's just to point out that there are a lot. I mean, we have 19 bat species in the UK. There's more in continental Europe. Some of these are all largely little insectivorous microbats. We only have one population of fruit bats in Europe, and that's in Cyprus. So we're really talking about little bats here. They often share roost sites. If you look at the phylogenetics of these viruses, it's quite confusing, mostly because most of these are just point studies, and mostly PCR point studies, because it's the easiest way to look at them is to go and stand under a bat roost with a sheet, with a tarpaulin sheet, and collect the poo as it falls. and this happens sporadically, but we don't do, there's pretty much no long-term studies of individual roost sites. Some of those are coming on board now. So it's really just a point estimate, and the prevalence rates vary from somewhere between 2 to over 42%. So it's quite wildly variable, and we've got a mix of different alpha and beta coronaviruses. None of the ones we have detected in Europe are very convincingly zoonotic. They, as far as we know, we mostly only pick them up in bats, but that is potentially because of a lack of study. So, what has been done in the UK, and this is work we did with BBSIC funding during the pandemic, is sub-eco viruses are the ones everyone gets very het up about, the SARS-like beta coronaviruses. And the natural hosts of these are this pretty much confined to horseshoe bats, which are these little cuties that look like very fat little Pokemon being monitored in their winter roosts down the bottom there. So what is important though here in this phylogenetic trees from a publication of ours, so the ones that are reported in Asian horseshoe bats are quite phylogenetically different to the ones that we see in Africa and Europe. So they're really distinctly geographically clustered, and probably also clustered by species, at least somewhat. The ones we have in Europe do not have that furin cleavage site that's thought to convey zoonotic potential, or at least cross-species infectivity potential. That's really only been detected in horseshoe bat populations in Southeast Asia or southern China. And there's some very particular epidemiology about the size of those populations and the numbers of species that cohabitate some really large roost sites that doesn't exist elsewhere. And that's probably a large part of why we see this difference. So that's what we're looking at in Europe with bat sub-ecoviruses. And this is from our UK study. So what we had in the UK was we looked at a variety of bats. We were focused on the two horseshoe bat species we have, and important to point out that these are both quite rare here. This is at the limit of their range. So we're talking about less than 20,000 animals in at most a couple of hundred roost sites for these species. Interesting, we picked up that a European bat sub-ecovirus, but interestingly, we only found it in one of the species, even though they cohabit the same roost sites, and we don't have a clue why at this stage. But we did sample quite a lot of animals, so we're fairly confident in those numbers that we should have picked it up. And there's nothing obvious in those numbers about age, sex, geographical distribution. We know nothing about excretion, whether it's more common in juveniles, adults, winter roosts, summer roots. We have no idea. This is all we've got at the moment. It's spread across their range. Not every roost site was positive, but quite a high prevalence rate. No association with disease that we or anyone else has ever seen, but if it causes, say, mild diarrhea in juveniles, which is quite likely for coronaviruses in animals, that's going to be really impossible to pick up in a wildlife species, a transient diarrhea that gets better. We're not going to see that. So, some more recent work, which some of you may or may not have seen. One of the other coronaviruses that we do pick up reasonably often in wildlife in the UK is mabicoviruses in hedgehogs. And this is work from Andrea Cruz, a PhD student from Portugal, which we're co-authors on, where they were looking at the coronaviruses present in hedgehogs, in rescue centres in Portugal. They picked up so one of the more interesting things about this is they picked up an alpha coronavirus that we couldn't retrieve full sequencing data from just because of the sample quality, but it was very, very similar to a virus that had been identified in bats in the same location. And I cannot tell you any more about it than that, but that is the limitations of where we're at with our depth of knowledge on these. They looked at with the hedgehogs. There's been quite a lot of study of the hedgehog mabicoviruses and quite like several hundred animals in different countries, and no one's reported a convincing association with disease. But as I pointed out with the bats, transient diarrhea in a wildlife species that they get better from is going to be hard to detect. It is very common. Prevalence rates, depending on the study, vary between 10 and 58%. And like with those horseshoe bats, when you're getting up to sort of 50% of your animals excreting it, that's probably an indication of a chronic excretion. And we do see that, say, with cats, with the enteric coronaviruses, but we don't actually know a lot more than this at this stage. So we did sequence these, and the guys in Portugal did some protein homology modeling because we don't know what the receptor for this is, and that needs to be done in cell culture studies, but this is as far as we've got at the moment. So the MERS receptor is a DDP4, and when we overlay the MERS and the hedgehog coronavirus structures, which is the diagram up the top, they're reasonably similar, not identical. But when we do protein docking scores, the scores for the hedgehog DDP4 and this virus are not great, not very convincing. There is a higher homology, at least on this in silico docking, for ACE2. And they also had a look at both human, and this virus looks like it would probably bind stronger to the human homologs of these than the hedgehog ones, and also the species that are most likely to eat hedgehogs. So cats, foxes, badgers, our major small predators in Europe. And in some cases, there's a reasonably high homology or binding chance for these with those predator species. So there's still a big question mark over what the actual cross-species transmission risk is, if there's any. We're still at early days with this. That cell culture work still needs to be done. And just to demonstrate why we worry so much about hedgehogs, the picture down the bottom is one of my chickens, and one of the hedgehogs in the garden had chosen to have a sleep in the hen nesting box for the day. So these are really common animals in and around habitation in urban areas in Europe, and a lot of them get handed into wildlife rescues. So we are in contact with them a lot, and so are our pets, our dogs, cats, and chickens, apparently. So more hedgehog work. This is another study from Gabriele Ratti, who's an Italian PhD student who came and did an externship with us as part of his European microbiology veterinary resident training. And he was looking at exotic species hedgehogs that are kept as pets in Italy. Gabrielle's very big on his exotic species. And he didn't find any coronaviruses in African pygmy hedgehogs. And there's one Japanese study that's found the same thing. No studies at all of these things in the wild. But he did pick up a Mabico in long-eared hedgehogs, which are a completely different species to the hedgehogs, the wild hedgehogs we have in Europe. And there's a little picture, the little cuties here. You can see why people keep them as pets because they are incredibly cute. And these long-eared hedgehogs are the important bit of this. If we come over and have a look at this phylogenetic tree over here, is the hedgehog viruses we've had reported previously, one in Chinese hedgehogs, quite a bit of study of the European hedgehog, the common European hedgehog. And these guys, where they're from naturally, and they're only fairly recently domesticated, are in the middle. So the Chinese hedgehogs are way off on the other side of China, European hedgehogs here. These guys are native to Central Asia or the Middle East, and their coronavirus plunks up smack bang in the middle. So it looks like we've probably got a kind of a continuum of these Mabico viruses across different hedgehog species linked to where they are geographically. Uh, we don't know any more about that as to how species specific these are or not. But this is what we've got at the moment. So what I find quite disturbing about some of this is these are really common animals, and we know this is all we know about them and their viruses. So moving on to rodents, which are, along with bats, the other most abundant mammalian order in Europe. Uh, we have done some previous study, and this was actually pre-pandemic, looking at alpha coronaviruses of European rodents. So all of these studies were done with pan-coronavirus screening PCRs, and this is what popped up. So we didn't detect beta coronaviruses in these. It's the same PCR we've used for the bat studies, so it should pick them up. But we do pick up these alpha coronaviruses, and these are more interesting than I appreciated at the time we found them. So these are one of the few really convincing examples of probable recombination across coronavirus genus, so between alpha and beta coronaviruses. However, this is not a recent historical event. This is a evolutionary event a very, very long way back in the evolutionary tree, because most, possibly all, of the rodent coronaviruses that have been identified in Europe, China, all are all very part of this same phylogenetic group with this probable recombinant pattern. Um, Interestingly, there are also a small number of other bat and one pig coronavirus, and these have now been called leukoviruses taxonomically. But we also see these so this study, we also picked them up in rabbits and shrews, which are not rodents. They're a different taxonomic group, insectivores. But we just have so little study in this group that it's really hard to tell actually is this a rodent virus? People write all sorts of things about, bats being the origin of all coronaviruses, but really we've done so little monitoring in other groups that who knows? So moving on to what we are doing, a bit of what we're doing now is so this is my current PhD student, Charlotte, who's here today, I think. We know that mink, ferrets, foxes, small carnivores are susceptible to canine and feline alpha coronaviruses because that gets reported sporadically. We have an issue with canine and feline alpha coronaviruses, the Tagaka viruses, where they do quite readily swap genes, and we have multiple recombinant variants, and we are seeing periodic waves of new variants appearing with altered pathogenicity. The most recent one, the feline coronavirus outbreak in Cyprus, which is spreading northwards as far as we can see. For those who know more about domestic animals, we also do see an FIP-like syndrome in ferrets, which that's, as far as we can work out at the moment with cats, this is probably a rare manifestation of more severe coronaviral disease in animals that are have problems with their macrophages that mean they don't control the disease very well. There's a constant argument as to how much of this is the cat's genetics and how much of this is the virus's genetics that results in these macrophage tropic viruses causing this severe disease. But we don't actually know very much at all about this in ferrets. So we've just retrieved a collection of archived ferret FIP cases and ferret coronavirus, enteric coronavirus cases from Glasgow, which Charlotte will be sequencing soon, and that will be really interesting to compare with the cats. Well, we did do quite a bit of work during the pandemic with the grant we were looking at, looking for SARS, we and other groups. This is why we've had such an explosion in this area. And we didn't find SARS, which was good, but we did find a novel alpha coronavirus in stoats on the Orkney Islands in Scotland. And probably unsurprisingly, it's clusters along with the ferret and mink coronaviruses that have been reported in captive populations before. But once again, this is a really, really common species in the UK, and I was a bit astonished that we're still finding completely novel viruses in them, but that's how little looking we do. And we also, off the back of the sequencing data we did with that, one of the advantages of just doing a plain old mRNA-seq without any enrichment is we can also have a look at what other things are in there and have identified also novel caliciviruses out of that data and those animals. And these are just wild trapped animals as part of an eradication program. So once again, we know absolutely nothing about disease. The ferret and mink ones are probably associated with diarrhea, with gastrointestinal disease, but that's as much as we know. So ruminants, probably we're not going to talk about pigs today because we don't have that many wild pigs in the UK, and they tend to share their viruses with the domestic ones, in which there's been a lot of study. We do, however, have quite the other major mammal group we have in Europe are ruminants: deer, goats, sheep, chamois, bison, red deer, etc. And this phylogenetic tree, which I know you can't read, but was done by a recent PhD student on a placement, Molly Simpson. I asked her to go and fish out all of the non-cow reports of coronaviruses in ruminants, and basically uh, all of the isolates she can find in the public databases cluster with the cattle isolates, so they are in essence variants of bovine coronavirus. And the interesting thing about those is they're scattered in and amongst the cattle isolates from the same geographic region. So what that looks like, there's very little data to really go on, is that these are probably constant spillovers and or shared back and forth with the cattle population rather than something that circulates separately in the wild ruminant populations. But there's been very little actual work done aside from a few zoo outbreaks. So this is another area where we've just not got very much information at all. So SARS, this is a little bit outdated now, but this is taken from a European Food Safety Authority review, which is probably as good a place to start as any for a summary of the huge and confusing variety of papers on what has and hasn't been detected when and where. But basically, the only really convincing circulation of SARS in a species outside of its original host range or people is the white-tailed deer in North America. Most of these other detections are detected sporadically, but we've not really seen convincing evidence of wider circulation. So that's what this is about. So we've had two exceptions to this in Europe, so to this sporadic detection of cases in wild animals, and these were both in fallow deer, in parklands in very densely populated cities. So the Dublin study has been published, and George Carnell, who's about to start with us at Nottingham, has some unpublished data which should be coming out soon, which looks rather similar with the population in London. And we have done a lot of looking elsewhere in the range of deer we have and not seen it. So why this difference within the USA and these two populations where it's looking pretty convincing that they've had Omicron infections, and it is with the Omicron variant. These are really high density populations. They're confined populations, both were fallow deer populations, and they're in heavy contact with people. So the picture there of someone giving the deer a drink from their Coke bottle this is what happens every day in these parks. It is impossible, despite having rangers patrolling the whole place and shouting at people and telling them off, to prevent people from trying to interact with the deer very closely. So that really heavy exposure to the infected human population is probably the big factor on why this spillover has happened in these populations and not in others. So what Charlotte is doing at the moment is focused on mustelids and continuing the sampling network, or trying to establish a sampling network for looking at these. So most of our small carnivores in Europe, but in the UK in particular, are mustelids. And we know they swap coronaviruses between each other, and we've had all those outbreaks of SARS-CoV-2 in farmed mink. So they are a big risk of a spillover. They also hunt and eat other animals, and fighting blood spread. This is a really high risk way for being exposed to any sort of cross-species transmission virus, and these guys are an important nexus of that cross-species transmission. So the diagram Charlotte's got over here just shows you what has been reported where and what species we've got and their numbers in the UK. So just so you can get an idea of what we're looking at. The methods are fairly straightforward, ones we've published before, ones lots of you use. We run a pan-coronavirus PCR, pick out the ones that are positive on that, sequence those with Illumina mRNA-seq, and then stick it back together bioinformatically to see what we've got, which is fairly crude, but it works. And when we're really not sure what we're looking for, and they're quite often quite divergent viruses, this tends to result in better coverage or retrieval of what we're looking for. So one of the ones Charlotte's been working on is badgers, and there has been recently a novel coronavirus partial sequence reported in Italy from a group there. But we've been unable to detect it in our UK badgers, and we've tested hundreds by now. We have sporadic positives that are perhaps potentially a bovine coronavirus related sequence, but because of the sequencing quality, we just haven't been able to confirm that or find anything that looks anything like the virus that was reported in Italy, despite the fact we're using virtually the same methods. And most of the badges we get in are from roadkill surveys coming through our post-mortem facility. So sample quality is an issue. But the numbers that have been tested by now by us and others, we really should have found something. So I'm not a big question mark still over that. But we're certainly not picking up what the Italians have found. And this is where we're at with other samples at the moment. So it's a long, slow process building up this sampling network. And we've now got a steady stream of things coming through. Charlotte spent a lot of time running around talking to wildlife groups, RSPCA hospitals, rescues, hunting groups. There's a huge number of small organizations, often charity organizations, who are actually the ones managing and interacting with the animals and have access to them. And there is no point going outside of those networks, but it involves a massive amount of work to actually maintain those relationships and keep talking to those people. And Charlotte's doing very well at this. So excitingly, we've recently had a positive in a fox from one of the RSPCA hospitals, and it looks like it's probably something related to the canine alpha coronaviruses. But we've just sent that off for sequencing, and we'll see what that is and where that fits, whether it's one of the dog isolates that we know we have circulating in the UK, or whether it's something specific to foxes. So that's really it. I'm hoping I've not run too over time. And there is a cast of thousands of rapidly expanding people involved in this, partly because of all those community groups that send us samples in. And I will stop that there.
Thank you. So thank you for sharing all this data. It looks a lot of sampling. So
Yeah,
that's
I sat
yeah.
down last night and went, that's a lot.
Yeah, that's a lot. It's not that intuitive as just a graph, right? It never is. So does anyone have any question? I don't think I can see any hands. So I had a couple. I don't work at all in this field, so maybe my question will be a bit well, just bear with me.
That's fine.
So
Charlotte was
when
presenting to the RSPCA vets the other day. So
so I was wondering because that's you show the sampling you got for bats you got in the UK, and you said for I think greater Horseshoe bat you couldn't find any coronavirus sequence, but you can find some in lesser Horseshoe bat, if I understand it
Mhm.
correctly. So I was wondering when we read. It's true that we don't know about, sometimes we not always know about negative data and publications. But I was wondering how can we explain or justify the richness in sequences that we find in other parts of the world, like in in Asia or in Africa. Like I have the feeling whenever they sample a bat, they found a lot of viruses. So do you have any explanation for that?
That is so there's probably two things going on. So Southeast Asia, they have an enormous biomat, an enormous number of bats. So those horseshoe bat roosts in the mountains between China and Vietnam can have 20, 40,000 animals in them. That's more than we have in the entire country. And that sheer density of animals facilitates virus transmission. And these are multi-species roosts with up to 6 or 8 species, horseshoe bat species in them. So that ecology of the population makes a massive difference to virus spread or not. And if you look at, say, the mink with SARS, we saw those massive, massive outbreaks in farmed animals, but we do not see it in wild animals. We just do not, because in the wild they are solitary. Uh, and it's when they're in those big population numbers together and there's lots of juveniles that really facilitates spread. So that population ecology is this has been a question all the way, like, why are we seeing this in some species, others? Why are we seeing it in the US deer and not in the UK and in the European ones? And I really think this has very little to do with actual physiological species susceptibility and much more to do with population density. Does
Okay.
that make sense?
Yeah, that actually, yeah, completely it does. And so I will ask a second question if no one wants to jump in. I was wondering, this is a bit aside, but we work a lot on, like me included, we work a lot on alpha coronaviruses and beta coronaviruses. And the gamma and delta are a bit forgotten. So recently there is this epidemics of flu going on, and so I was wondering if maybe because of this, because the awareness that was raised during the pandemic, there is more of a willing to share resources, and if you get access to birds, like samples of birds, and then you could, I mean, I understand you're already doing a lot on mammals, but I
Yeah.
was thinking, oh, maybe this
I
is
mean,
easy access
me
because people have
personally,
samples.
I have to draw the line somewhere with the number of
Sure.
things we're doing. And curiously, the H5N1 situation in birds has probably actually made it harder to get people to give you samples to run agnostic screening on, like this kind of thing, because nobody wants to report a notifiable, be caught holding a sample that's a notifiable disease.
Mm.
Uh, so the RSPCA hospitals in the UK, when they did report a H5N1 case in the birds, the entire hospital was shut for more than 6 months.
Oh,
So
yeah.
they're not going to give us bird samples to run agnostic screening on because they
Okay.
know that H5N1 is rampant
Yeah.
in the wild population, and
Okay. Mm.
it's actually made things worse.
Oh,
And in
okay.
theory, we could, like, if and all the reporting of H5N1 sequencing is all enriched for H5N1. So there is no data that's not producing a bonanza of other data to harvest,
Okay.
unfortunately.
Yeah. Okay. Well, I think for sake of time, we can move to the next speaker if no one has any question, or you can just, I guess you can email
You can
Rachel if
always
you have.
email me or
Yeah,
Charlotte.
exactly.
Or,
If
yeah.
something comes to your mind.